So yes, now we are going to discuss pathogenesis of cancer. So the pathogenesis of cancer, this is a normal picture indicating a normal cell, which under these were the etiological agents of cancer like chemical carcinogens, radiation, viruses. So under these etiological agents, which when they affect on this normal cell, they causes DNA damage. And when this DNA damage is permanent and it is unable to return to normal, then it will be converted into mutated cell. So thereby genes affecting apoptosis, it will be converted into mutated cell, and because of activation of oncogenes and inactivation of anti-oncogenes will counteract these oncogenes. So inactivation of anti-oncogenes and activation of oncogenes and apoptosis regulating genes, that means which will prevent this normal cell death. So under these conditions, this cancer cell will be converted into malignant tumors. This is a general overview of this pathogenesis of cancer. So depending on this etiology, this pathogens of cancer is explained under four topics, like molecular pathogenesis of cancer. That means here it includes genes, genetic genes which will regulate this which are involved in the pathogenesis of cancer. So genes and cancer, that will be studying under molecular pathogenesis of cancer. And chemical carcinogens and chemical carcinogenesis. So how does this chemical carcinogens or the chemicals cause cancer, that will be studying here. Physical carcinogens and radiation carcinogenesis, biological carcinogens and viral oncogenesis. So under these four, we'll be studying this pathogenesis of cancer. And coming to molecular pathogenesis of cancer, so it is also called as genetic mechanism of cancer. Okay. So it will be it includes what does it include is it includes monoclonality of tumors. That means what are the characteristics that are included, or like monoclonality of cancer. So that means a clone or group of cells will be considering as clone. So that monoclonality of tumors means that group only are capable of undergoing replication. So most human cancers arise from a single clone of cells by genetic transformation or mutation. Among all the cells, only a few clone of cells undergo genetic transformation and mutation, and they will be converted into cells. And field theory of cancer. Field theory of cancer, it includes in an organ developing cancer, that means in organ in which cancer is there. In the backdrop of normal cells, limited number of cells only grow into cancer after undergoing sequence of changes under the influence of etiological agents. That means if a particular organ which is having cancer, in that all the cells doesn't have this cancer nature or tumor characteristics, only a few or limited number of cells that will grow into cancer cells after undergoing sequence of changes, that means sequence of mutations, so by under the influence of etiological agents. So that is field theory of cancer. That means only a few cells will be involved in the development of cancer. And multi-step process of cancer growth and progression. Usually it's not a single step, it involves multiple step process, like depending on different steps were involved, like initiation, promotion, that will be studying the next class, like in the next slides. The various causes may act on the cell on one after another. So first one step will take place, followed by next step. So it includes many number of steps, that we call it as multi-step process. The same process is also involved in further progression of the tumor. So this multi-step process is involved in the initiation of tumor and also in the progression of this tumor. So this is all about this. So monoclonal origin of tumor cells in uterine leiomyoma. So G6PD allele A, G6PD allele B. This is the normal adult smooth muscle cells. So type A and type B, A and both. So type A, this is normal myometrium. Type A and type B. So monoclonal origin of tumor cells in their leiomyoma. So generally it should be in this way, but because of some mutations in these genes, it causes leiomyoma of uterine muscles. And genetic theory of cancer. In cancer, there are either genetical abnormalities or there are normal genes with abnormal expression. That means either the cell may be having genetical abnormalities or during the process of expression, so it may have abnormal expression. So these abnormalities in genetic composition may be from inherited or induced mutations. So it may be inherited or it may be induced mutations. Induced mutations can be due to any carcinogens like physical carcinogens or chemical carcinogens or any virus. So that we call it as induced mutations. So this whatever the mutated cells are there, they will transmit their characters to the next progeny of cells and result in cancer. So from one cell to other cell that gets transferred and will result in cancer. And then general regulators of normal and abnormal mitosis. So activation of growth promoting oncogenes causing transformation of cell, inactivation of cancer suppressor genes, abnormal apoptosis regulatory genes, failure of DNA repair genes. So first is activation of growth promoting oncogenes. That means these will promote the growth of tumor cells. So they will be activated. And inactivation of cancer like anti-oncogenes. That means which will suppress the growth of these tumors. So that genes will be suppressed. And abnormal apoptosis regulatory genes. Usually for every cells they should undergo apoptosis after a particular lifetime. But that will not be taken place in tumor cells. That is due to abnormal apoptosis regulatory cells genes. And failure of DNA repair genes. Usually if there is any damage to the DNA, there are certain particular genes which will repair this damaged DNA, but that also will be failed. Failure of DNA repair genes. All these may lead to formation and progression of tumor cells. So once again I'll repeat, monoclonality of tumors, that means only a particular clone of cells will undergo replication or genetic transformation and will be converted into tumors. In field theory of cancer, only in a particular organ, only a few cells will undergo replication under the influence of etiological agents. Multi-step process of cancer growth and progression. So for the cancer growth and for the progression of tumor cells, it is not taking place in a single step. Multiple steps one by one will be followed. And genetic theory of cancer, here what will be there? Either there may be abnormality in the cell, genetical abnormality in the cell, or there may be there may be abnormalities in the expression of the gene. So either of these two may take place, and these abnormalities may be either from inherited, from birth, if it is like from birth, or induced mutations. Induced mutations is nothing but under the influence of any carcinogens or cancer etiological agents, and with the under the influence of that, if these mutations takes place, that are called induced mutations. Okay, so once these mutations were done in these genes, so they will transmit their characters to the next progeny of cells. So thereby, so this cancer kind of nature will be transferred to the other cells, resulting in the cancer. And genetical regulators of normal and abnormal mitosis. Here there will be activation of oncogenes and inactivation of proto-oncogenes like anti-oncogenes, that is nothing but cancer suppressor genes will be inhibited, and abnormal apoptosis regulatory genes and failure of DNA repair genes. So these were the main things that will be taking place. If you see this, so it's a representation of major properties of cancer in terms of molecular carcinogenesis. Excessive and autonomous growth. First is excessive growth, and second is refractoriness to growth inhibition. That means inhibition will not be inactivated. That means there will be continuous growth. Escaping cell death by apoptosis. So apoptosis will not be done because of abnormal apoptosis regulatory genes. And avoiding cellular aging. Generally cells undergo senescence, that will be avoided. Continued perfusion of cancer. Perfusion of cancer means that will be spread to enter into these capillaries and will be spread to the surrounding tissues. And invasion and distant metastasis, it may spread locally or it may spread to distant places. DNA damage and repair system. So DNA damage will be there, but repairing will not be done properly. So because of that, mutations will be taking place. Cancer progression and tumor heterogeneity. So there will be progression of this cancer. That means it will be spreading, spreading, and multi-step molecular phenomena. So these steps takes place one by one, and microRNAs in cancer. So RNAs, as they are genetically mutated, their structure is also altered. So these were the things which were going through in the process of molecular carcinogenesis. Okay, once have a look over this. Excessive growth, growth inhibition, escaping cell death by apoptosis, avoiding cellular aging, continued perfusion of cancer, invasion and distant metastasis, DNA damage and repair system, cancer progression and tumor heterogeneity, multi-step molecular phenomena. So these were the properties of cancer in terms of molecular carcinogenesis. So so how these proto-oncogenes were activated to form growth promoting oncogenes. So if you see this, these were the growth factors like PDGF beta, TGF alpha, FGF, HGF. So these were the receptors for these growth factors. So these were the growth factors, and these were the receptors of these growth factors if you consider this as a cell, nucleus and DNA. So cytoplasmic signal transduction proteins, so mutated RAS, BCR, ABL, like BC receptor, ABL. So under these signal transduction proteins, nuclear transcription factors, they gain access into the nucleus. When these nuclear transcription factors include CMYC, NMYC, LMYC, cycle regulatory proteins. So these were the cell cycle. So because of this, there will be abnormal or mutations in this nucleus. In, so it includes cyclin D, cyclin E, CDK4. So these were the, so by this way it will be converted into growth promoting oncogenes. So first this oncogene gets activated and will be converted into growth promoting oncogenes. So these were the important oncogenes, mechanism of action, and what are the different types of tumors. If growth factors like PDGF beta, TGF alpha, FGF, HGF, so these were the growth factors. So the proto-oncogenes here include SAS, RAS, Hist2, INT2, HGF. These were the proto-oncogenes to their corresponding growth factors. Mechanism includes overexpression, amplification. That means overexpression of these proto-oncogenes. So when this PDGF beta is overexpressed, that is proto-oncogene SIS is overexpressed, it causes glioma, sarcoma, carcinoma, sarcomas, bowel cancers, breast cancer, follicular carcinoma, thyroid. So under the influence of these growth factors, these proto-oncogenes will undergo overexpression and leads to these tumors. And then receptors for growth factors. So about this we have seen now. Now these receptors for growth factors: EGF receptor, c-KIT receptor, RET receptor. c-KIT receptor is also called as steal factor. So under this, the proto-oncogenes include ERB-B1, ERB-B2, c-KIT, RET. So again here, this is the overexpression or amplification. For these two, there is point mutations. So this causes squamous cell carcinoma. So when these proto-oncogenes were reacting with these receptors for growth factors, when they undergo overexpression point mutations, so the result will be glioblastoma, CA breast, or CA ovary. That is, CA is nothing but carcinoma, ovary, stomach, lungs. So these will take place. And cytoplasmic signal transduction proteins, GTP bond here, RAS oncogene, when it undergoes point mutation, it causes causes carcinoma of lung, colon, pancreas. Non-receptor tyrosine kinase here, the proto-oncogene is BCR-EBL. When it undergoes translocation, it causes acute leukemia. Nuclear transcription factors like CMYC, NMYC, IMYC in the proto-oncogenes, when they undergo translocation or amplification, again they causes these kind of cancers. Cell cycle regulatory proteins like cyclin, CD kinase, So when they undergo translocation over expression or amplification, these kind of cancers will be seen. So if you see this, so these were the factors which were involved, and these are the proto-oncogenes which were released. And when these undergo overexpression, amplification, or point mutations, translocations, so when these undergo these kind of mechanisms, these were the different types of tumors that will be occurring. If you see this, what were the differences between oncogenes versus anti-oncogenes? So oncogenes were derived from mutated form of normal proto-oncogenes. So when these normal proto-oncogenes were mutated, then oncogenes were formed. So anti-oncogenes were formed when mutated form of normal growth suppressor genes, genetical abnormality. So when it undergoes mutations like point, translocation, amplification, overexpression, retroviral insertion, and DNA damage. Here, loss of genes by depletion, point mutation, loss of portion of chromosome. Major action includes allows cell proliferation by increased growth promotion pathways. Here, anti-oncogenes allows cell proliferation by removal of cell growth suppressor pathway. By removal of cell growth suppressor pathway. Level of action in cell at different levels, it includes different activities. Here at different levels, like it acts both at cell surface, cytoplasm, and nucleus. So major types of oncogenes include growth factors, growth factor receptors, cytoplasmic signal proteins, nuclear transcription proteins, cell cycle regular proteins. So all these were the types of oncogenes. Then coming to anti-oncogenes, HRB, P53, TGF beta, APC, and beta catenin, and others like BRCA, VHL. So these were the examples of anti-oncogenes. So if you see this picture, these were the apoptosis associated genes. These were the anti-oncogenes. So these were transcription proteins like MYC. These were DNA repair genes or mutator gene. This is telomerase. These two were the cell cycle activator inhibitors. So this is all these were present in the cell nucleus. This is the cytosol. This is the cell membrane. So these were the receptors for growth factors, like, and these were the growth factors like VDGF, TGF alpha, FGF. These were the growth factors. These were the receptors. These two comb-like things were signal transduction proteins, so which will helps in the transduction of signals from outside to this inside of the cell. So if you see this, when growth factors under their influence, when they bind to these receptors, they gain access into the cell, and they will pass this information through signal transduction proteins, and this is the DNA. So these were the different types of genes which will act upon and will cause the mutation of these cells and will give it a characteristic tumor like nature. This is all about molecular pathogenesis of cancer.